セールス&マーケティングダイレクターのスチュワート・ケネディさんです。がん診断におけるカスタムメードFISHプローブの開発。
Good morning, ladies and gentlemen. My name is Stewart Kennedy. I'm the Social Marketing Director at Cytocell. As you mentioned in the first speaker this morning, you heard about the Cambridge cluster. Well, we're one of the early Cambridge cluster, I would say. So, Cytocell, we've been going for over 20 years. Our business model has been purely based on fish, and fish has been around for just over 20 odd years. Within that area, we started off looking at constitutional cytogenetics, so we're looking at genetic aberrations, and as developments have gone on looking at genes and where genes are important, and finally getting into the oncology side of things, we've progressively moved our business model with that. So we moved from the constitutional cytogenetics into then look at leukemias, and from leukemia we've then moved on to solid tissue. What I wanted to talk to you about is really our MyPros concept, which is our own way of developing new products for ourselves, but also to assist the researchers out in the field as to how they can source fish probes very quickly and very easily. So the way we do this is we actually obtained our own BACS collection, and within that collection we have over 225,000 clones that are owned by ourselves. Our clones, with these we know where they're in sequence, so we know where they sit on the human genome. So we have our own copy of Ensemble. From Ensemble we can look on by coordinates or by gene name as to what target somebody may be interested in, and then be able to select the clones that would produce the product for them. So there are three real project sizes that we have, and over time, since we started offering this as a service, we've done over 100 different projects. Now these can range from anything being from the very small scale, where we may do 50 tests for someone and they want to do initial research into a gene and see what interest there might be with it, right the way through to we do, for one customer in the US, 10,000 tests in one batch at any one time. So the scale can be moved depending on what your needs are. So the complexity side of things, a small project, this is really just taking a routine catalogue product and maybe changing the fluorophore, so the dyes that we use with it, and maybe using a different control probe with it. A medium project, this is where things start to get more interesting. Here we're actually looking at the researcher telling us, or company, pharmaceutical company, turned around and saying, here's the target we want to identify, and from there we actually build a brand new product that is not on the market or available from any other source. And then the final stage is a large project. Here, this is getting into a very complex format. So we're talking that we could have up to 5 different fluorophores, so you're potentially looking at 5 different genes in one particular test. So the process, there's really 5 stages to the process that we do to ensure we produce the correct product for the customer. The first stage with this is really finding out from the customer what do they want, and then secondly for us to go back and ensure that we have understood their requirements and discuss with them maybe some pitfalls that they have in their strategy, or concerns that we may have or they may have as to what the design of the product's going to be. Then we go into developing it, optimize the product itself, produce the product, and finally do the QC release. The format that we typically do, these are either, we produce them under the same criteria as we do for our FDA ASR, analyte specific reagent, or CE marking. And then they typically have the same format as what you have for a standard catalogue product that we produce, so the storage conditions and shelf life. So the first stage, is the customer telling us what they want. So identifying what gene it is, how the product should be built, the strategy behind that, and what number of tests that they may require. Then we go back and actually return that information to them. Part of this is to actually show a copy of our copy of Ensemble with the target gene on it, and then what clones we've selected for it, what fluorophore they want them matte to be coloured in. So here we have an example. We were the first people to do an EML4 probe several years ago. So we had a request for this from a US laboratory. From here you can see the target, which is circled in black, being the EML4 gene itself. And then below it you see the two different clone areas for this to be a break-apart probe. So the green section is the clones that we selected for the green half of it, and red being the clones that we selected for the red half of the probe itself. So then once we've agreed all of this, and both ourselves and the customer understand what we're actually going to produce, we go into a typical R&D setting where we develop small scale sets, ensuring that the product's actually going to work, addressing any concerns, and once we've optimised that, then we move into full-scale production. So within our production of the probe, we treat this, even though it may be done as a custom product, we treat it the same way as we do any standard catalogue item. So it goes through the same quality procedures as we have for the rest of our business. We are both ISO 9001 and 13485 compliant, as well as GMP and GLP. So the final stage, once we've actually produced it, is to do the QC release. So again, we treat this the same way as we do with our catalogue products. We would fish the actual kit itself on cell samples, so depending on the sample of choice, this could be lymphocytes, it could be bone marrow samples, it could be FFPE, whatever the customer wants to use the kit for. So in conclusion, MyProbes is development of unique fish probes based on customer specification. It utilises Cytocell's extensive BACS collection. To date, we have over 100 projects that we've supplied, and we see this as being really the forefront of companion diagnostics. Thank you for your time. Any questions, then I can answer them.